A Sweet Representation of Mitosis and Meiosis by Emily Vallejo and John Dumay. An organism's ability to reproduce its own kind is the characteristic that sets living things apart from non-living matter. Mitosis and meiosis are important because they enable cells to reproduce, grow and develop, and renew tissue. These processes are the forces that drive life itself.
Interphase. There are three stages of interphase: G1, S, and G2. A cell grows in G1, continues to grow and copy its DNA in S, and grows even more in G2 as it completes preparation for cell division. Three checkpoints occur which ensure that the cell meets the requirements for mitosis. DNA is loosely folded, and two centrosomes are formed, each with a pair of centrioles.
Prophase. As the cell enters prophase, chromatin condense and become tightly coiled, forming loose chromosomes. The chromosomes consist of two sister chromatids joined by centromeres. The nuclear membrane begins to disappear. The mitotic spindle composed of centrosomes and microtubules begins to form. The centrosomes begin to move to opposite ends of the cell and are driven by the extending microtubules between them.
Metaphase. Metaphase is the longest phase of mitosis. Centrosomes are now at opposite ends of the cell. The chromosomes line up along the metaphase plate, and the sister chromatids are connected by kinetochore microtubules.
Anaphase. Anaphase is the shortest stage of mitosis, lasting but a few minutes. When the cohesion proteins of the centromere are split, anaphase begins, and daughter chromatids are drawn to opposite ends of the cell due to the breakdown of microtubules at the kinetochore. The cell becomes oblong as non-kinetochore microtubules lengthen.
Telophase and cytokinesis. In telophase, two daughter nuclei form around the separated genetic material. The chromosomes become less condensed. Nucleoli reappear. The M phase is complete. Once the components of the cells are fully separated, a cleavage furrow forms composed of a contractile ring of microfilaments. This process, which results in the formation of two identical daughter cells, is called cytokinesis. Each cell is diploid, meaning it has a value of 2 N chromosomes. Cell division is now complete.
Meiosis. As in mitosis, the replication of genetic material occurs prior to the beginning stages of meiosis 1.
Prophase 1. Contrary to what happens in mitosis, in prophase of meiosis 1, replicated homologous chromosomes exchange segments in a process called crossing over. The points at which this occurs are called chiasmata. As in mitosis, the nuclear envelope breaks down and the spindle fibers begin to form.
Metaphase 1. Pairs of homologous chromosomes line up in the center of the cell along the metaphase plate. However, unlike mitosis, two pairs of sister chromatids form tetrads and attach to kinetochore microtubules. The orientation of the homologs is important for the genetic variation of the daughter cells.
Anaphase 1. Homologs separate and sister chromatids remain attached, while they separate in mitosis. Similar to the process of mitosis, homologs move to opposite ends of the cell, guided by kinetochore microtubules. Non-kinetochore microtubules elongate the cell.
Telophase and cytokinesis. Unlike mitosis, daughter cells are haploid, meaning that they have one-half the number of chromosomes that were present in prophase. The nuclear membrane reappears as genetic material has fully separated. Each chromosome consists of two sister chromatids. Cytokinesis occurs in the same manner as in mitosis, with the formation of a cleavage furrow.
Prophase 2. Genetic material does not replicate prior to meiosis 2. In prophase 2, the spindle apparatus forms and the nuclear membrane begins to degrade.
Metaphase 2. Similar to mitosis, the chromosomes are lined up along the metaphase plate. Each sister chromatid is connected by the kinetochore to microtubules. Different from meiosis, sister chromatids are not genetically identical due to crossing over. The spindles move to opposite ends of the cell.
Anaphase 2. Just as in mitosis, the proteins holding sister chromatids together at centromeres are broken down, allowing sister chromatids to separate towards opposite ends of the cells, controlled by microtubules.
Telophase 2 and cytokinesis. Nuclei form around the haploid sets of genetic material, and the chromosomes begin to loosen. Cytokinesis occurs in the same way as in mitosis and meiosis 1. At the end of this cell division, 4 daughter cells are produced. Each cell is haploid, meaning it has a value of N chromosomes due to the lack of replication prior to meiosis 1.